Okay, so hello, fellow brain cells. Welcome to the very first episode in this week of in Neuroscience. I'm so excited to kick off this journey with all of you. Each week we'll dive into live neuroscience paper reviews, breaking down complex ideas, asking big questions, and making sense of the science behind the brain. Whether you're a student, a researcher, or just deeply curious, you belong here. Got a paper you want featured? Scan the QR code on screen and drop it in the suggestion forum. And this is just the beginning, and I can't wait to grow this space into a hub for learning, exploring, and connecting to science. So grab your favorite drink, get comfy, and let's spark some neurons together. So today we are going to be diving into risk factors for Alzheimer's disease and cognitive function before middle age in a US representative population-based study. Here I've got my highlights. You can ask any questions whenever you wish. I have also some streams notes that I'm going to share with you from today, and you can use this to guide yourself if you're new here. Okay, so uh, let's do. Looks like new papers have this new section called research in context, where they examine like evidence before the study and added value of the study and implications of all the available evidence. So the evidence before the study and the main thing about the study is that the majority of existing research on risk factors for Alzheimer's disease has focused on study samples of individuals 50 years of age and older, and there is way too little research on risk factors for this disease prior to midlife, and this presents a significant gap in our understanding. And here the authors looks like did a comprehensive search of PubMed and Google Scholar between these dates to identify studies examining key risk factors, uh, which include immune biomarkers, biological biomarkers, and social. I think they also examined. And here are the search terms if you're interested. But of course, once again, they reiterate that none of the reviewed studies examined all the risk factors in a single population as assessed here, and they found no population representative studies with a comparable age range. So, uh, the added value of the study is that they systematically examine these risk factors within healthy individuals of 24 to 44-year-olds. So they identify one of the earliest associations between key risk factors and cognitive function patterns. They use 3 different cognitive tests here, but we're gonna get to that. Um, and they found, unsurprisingly to me at least, that risk profiles emerge way before middle age, revealing early manifestations of cognitive patterns that may predict the decline typically observed in older populations, uh, which is quite good. So, yes, this is where they emphasize the need for early monitoring of risk factors such as cardiovascular, immune, ATN, ATN, which ATN are markers for neurodegeneration. Yes. Um, so they claim that the potential impact of their findings is significant, providing clinicians and health researchers with a clearer understanding of the early emergence of Alzheimer's disease risk factors before middle age, and laying the groundwork for earlier detection and prevention approaches. so the essentially what this study is saying is okay, listen, the risk factors emerged way before that. We could start detecting and treating. Wait, that's eventually what we want to invent our treatments that could be preventative. Preventive, sorry. Mm, okay, so yes, the risk factors encompass social, behavioral, clinical, and biological factors. And on the social and behavioral side, they are such as lower educational attainment, physical inactivity, and smoking. And clinically, health conditions such as diabetes, obesity, hypertension, and high cholesterol. Hypertension is essentially high blood pressure, and high cholesterol identified as consistent contributors to Alzheimer's disease risk. Also, a huge thing that they used here is this cardiovascular risk factors, aging, and incidence of dementia, or CAIDE, let's say, score is among the most, and this is among the most extensively researched risk algorithms and integrates the social, behavioral, biological risks. Every aspect of these risk factors. Okay. Let me check. Yes, including education, sex, age, cholesterol levels, blood pressure, BMI, and physical inactivity. And on the biological side, there are genetic, neurological, immune, and inflammatory molecules that have been uh, that that have been not detected necessarily, but they have been proven to be risk factors. One of those is a polyprotein E, which I learned now it's essentially a gene coding this polyprotein. Is we're going to dive into that a bit later. I have highlighted other things. Also, the amyloid, tau, and neurodegeneration, which is known as the ATN, biomarkers are increasingly promising. But again, in older populations, because that's what all the research up until now is about, as they say. And two commonly measured biomarkers are total tau, which is a type of protein, and neurofilament light chain. I searched what a filament light chain is. Let me just search it again because I completely forgot what it is. Neurofilament light chain. Yes, okay, Journal of Neurology, Neurosurgery, and Psychiatry. Neurofilament light chain is a neuronal cytoplasmic protein. Okay, so it's a protein highly expressed in large caliber myelinated axons. Its levels increase in the cerebrospinal fluid and blood proportionally to the degree of axonal damage in a variety of neurological disorders. Okay, good. So essentially it's indicator of the degree of axonal damage during some neurological disorders. Okay, that's good to know. So I've added some key questions and some key highlights by section I have for introduction, methods, and the results and discussion, sorry. It's quite short. I think short and sweet is what I wanted to make it. But let's get back to it. So let's see these. The two most studied markers are interleukin 6 and tumor necrosis factor alpha. I think tumor necrosis factor alpha speaks for itself. What it is, it's quite self-explanatory. But interleukin 6, I am not sure what that is. Let's explore what interleukin 6 is. Okay, National Institutes of Health. This PubMed is a really good source. This is verified by teachers and like academics. I've been told that it's a great source, so that's why I'm using it. Okay, so interleukin 6 promptly and transiently produced in response to infections and tissue injuries contributes to host defense through stimulation of acute phase responses, hematopoiesis. Mm, I would I would Google what hematopoiesis is because I don't know what that means either, but we'll get too far into the rabbit hole and we could go on till tomorrow like that. So yeah, the take home message is it's produced in response to infections and tissue injuries. That's good to know. So another marker of injury and infection as well. So mm, they assessed the associations between the KIDE score and the polyprotein E4 status to amyloid tau and neurodegeneration biomarkers, total tau and the neurofilament light chain, and several inflammatory molecules and interleukins, such as this HSCRP, highly sensitive C-reactive protein, and these are the interleukins and the tumor necrosis factor that we mentioned. With three standard measures of cognitive function this is important to point out that this is all in the US. The entire sample is US is representative of the US, but I think when talking about neurodegeneration, we might have more of a greater ability to generalize, at least in my opinion. I don't know what do you think? I'm curious to know what you think. And so the study population, it's data from waves 4 to 5 of the National Longitudinal Study of Adolescent to Adult Health, or for short ADD Health, were used for study and analyzed cross-sectionally at each wave. Now, what the heck does this mean? Oh, the waves 4 and 5 are different stages of this longitudinal study, in which the first wave, which was conducted in 2008, the participants were 24 to 34 years old, and this included it was a huge test that included many measures of many factors. Included in-home interviews, cognitive tests, physical exams, saliva collection, dried blood spot collection from all eligible respondents. And wave 5, which is the next one, conducted between 2016 and 2018, when participants were aged 34 to 44, and it was a mixed mode survey. This is important to note. It was a mixed mode survey combining in-person and web/mail surveys with a target out of which with a target of 12,300 survey participants, out of which only 1,702 received in-home interviews, which is not so good. Not so good, to be honest, but still, the sample size is still large. This must be noted. It could have been way larger, as we see here, but it's 1,700 is still quite good, from at least what I've been told by teachers. I'm not making this up on the spot. Mm. So, and yes, something else to notice is participants who reported being pregnant were removed from the study, and the wait, let me now we're on to the measurements. Let me just scroll like that. Okay, and we already talked about the KD score version. There are two versions of this. I forgot to mention. Two versions of this KD test. One, the only difference is that version 2 includes this polyprotein E4 status. Now, what does this APOE E4 status even mean? I wondered the same thing, and apparently, this status, in order for it to be positive, considered positive, they define it as having at least one E4 allele. Which, how it works, there are three different alleles: E2, E3, and E4, and E3 is I think the most neutral, harmless, and E4 increases the risk of Alzheimer's. This is proven. And why do we have E4 slash E4, E2 slash E3? Essentially, one of the E's is one E for each from each parent. So let's continue. You can see, I'm not sure if you can see actually, um, the way they measured this, these risk factors. If you want, you can pause. Okay, so they used the digital enzyme linked immunosorbent assay, which ELISA. Serum samples were saved for neurofilament light chain and plasma samples for total tau. Now, if you're like me, even though I'm studying for a minor in cellular biology, now it's going to be my major, I'm changing majors. This ELISA thing doesn't mean anything to you, so we'll look it up, see what exactly it means, what it means exactly. Okay, StatPros, this should be a good source. Let's zoom in a little bit. So enzyme immunoassays AIA. I don't know what's going on with me today. Use the catalytic properties of enzymes to detect and quantify immunologic reactions. Enzyme-linked immunosorbent assay is a heterogeneous EIA technique used in clinical analysis. In this type of assay, one of the reaction components is non-specifically absorbed or covalently bound to the surface of a solid phase, such as microtiter. How is this pronounced? Microtiter. Well, a magnetic particle or a plastic bead. This attachment facilitates the separation of bound and free labeled reactants. Oh my gosh. I am still confused, but as far as I understood, it's something like they, through an enzyme that recognizes this molecules from these immunologic reactions, they measure the immune response. As far as I know, as far as I understood. Okay, so values, looks like values were log transformed and then standardized. So I think that meant that they were turning them to z-scores. Missed. Okay, about the three cognitive assessments, These three cognitive assessments included immediate word recall, delayed word recall, and backward digit span, which examines the working memory. It's a measure of working ability of your working memory. So the procedure is essentially participants are read a 15 word list for a word recall and asked to recall as many of the 15 words as possible. Cognitive scores were standardized, yes, into a Z-score, as I said, for analysis, with a higher score indicating higher memory cognition. So, about covariates. Early life economic status apparently was operationalized using the social origin score. I don't know what that is as well. Let's look it up. Social origin score. Social origin. Let's see, this should be SPAC. This is an entire book. Social origin score. Let's see this. Ah, there we go. This cannot. Let me just read for a second. We measured social oranges. Oh, it's the same adolescent and adult health study. Members from information about their families collected from the study's participants at the Wave 1 interview, when participants is still or in middle or high school. We examine reports of parental education, parental occupation, household income, and household receipts of public assistance. These four measures were correlated weekly. That's quite a weak correlation, but okay. We conducted principal components analysis of the measures to produce a factor score, and the first principal component is playing 53% of the variance. We used loadings on this component to compute the social ranges factor score. Values are transformed to have mean of 0 and standard deviation of 1 for analysis. Okay, so it's essentially a report of these four factors. Okay, good to know. Good to know. So, additional covariates include age at exam. I think this would be easier if I just zoom this in a little bit more. Okay, so additional covariates include age at exam, educational attainment, at the same wave in which the respective biomarkers were measured, sex assigned at birth, smoking status defined by never versus past versus current, and an indicator for recent inflammatory conditions. There are probably these interleukins come at play. A race ethnicity variable was constructed from the Wave 5 survey and supplemented with Wave 1 data if missing. Okay, that's fair. We're going on to the analysis. So to examine the cross-sectional associations between the CAIDA score and this neurodegeneration marker, immune and genetic risk with cognitive functions, survey weight linear regressions were used to estimate beta parameters for unadjusted and adjusted models with each independent variable predicting each participant's cognitive score in the same wave. If you're like me, you are just really confused by this whole sentence. I was. It took me some time to figure it out. To be totally honest, but hey, we're all beginners here. Everyone was a beginner once, right? Mm, so, what survey way linear regressions mean? Survey way means that mm, because, for example, if most of the sample is from white people and uh, other races do not get nearly as well represented, then uh, they try to manipulate the weight of individual data points, so that the so so as to mimic sort of uh, representation, to mimic proper representation. It's not perfect, but it means they consider their um, take these variables into account, and mm, they estimate these beta parameters, which are sort of like a correlation. You can think it sort of like the R, Pearson's R. Okay. What was So there were known to results. There were no associations between having at least one E4 allele from the polyprotein E and cognitive function in both waves of the study. Mm, this is, I think, the entire set of markers. And here you can see the abbreviations. Okay, so we see the factors in the KIDE score here. It's a weight sum of education, sex, age, total cholesterol, systolic blood pressure, body mass index, and physical activity. In version 2, as I said, includes the same variables and only adds the polyprotein E4 carrier status. So now these are really important figures. This is the gist of it. This is both kind of versions, and these are the three cognitive test scores. They essentially hover around the minus 0.05 mark, which is, in neuroscience at least, a significant negative correlation. In both versions, and the unadjusted and adjusted ones are quite similar. Adjusted, I forgot to say what adjusted means. Adjusted, now you can see on each of the figures, in the description you should see what the models are adjusted for. And in this example, they're adjusted for race, ethnicity, social origin score, and indicator for recent inflammatory conditions and smoking status. So they essentially take all of those into account uh, statistically while doing this, while doing these analysis. Now these are the lines are estimates in where the result would lie and where the actual result lies. As on the wave 5 we see weaker correlations for the adjusted models, but still they do get quite close to minus 0.05. So there truly is something there that, how do I say, there is really something there that they've touched on. They have discovered something. It's not um it can't be said that the entire set of risk factors matter in people with ages 24 to 44, but they did discover that it matters, that your biomarkers at that age matter, even if not all covered by these different scores and tests. Plus, I think it's safe to say that I think this would have been better if they would have done more cognitive tests. I think that when it comes to cognition, three tasks are maybe a bit too low for a bit too little to truly measure cognition if we're talking about that cognitive ability. Uh, but again, they are well chosen. Also, I think. Mm, these are of course, they always have a 95% confidence interval, which essentially means that um, they their results would be true 95% of the time. This is essentially what the confidence interval is. These are some results that I've highlighted. If you want me to send the full full highlighted paper, you can ask me in the chat after this is uploaded, and I'll give it to you if it's any good for you, in your opinion. And now about the polyprotein E status, we can see that uh, for delayed recall and backwards digit span, there is clearly no statistically significant correlation. On wave 4 and on wave 5, the only thing we see is some positive correlation on immediate recall, but nowhere near not thing. Actually, no, my mistake. It's around 0.1, which is wow, which is actually interesting. I wonder if that's just a result from randomness. That's interesting, actually. Type type down below if you can what you think about this. Okay, so these were adjusted for sex assigned at birth and age, essentially, but they are like almost identical, mostly identical, both the unadjusted and the adjusted. So it doesn't matter that much. For now, whoops. So this is the between the ATN biomarkers and cognitive function. There is only no, forgive me. There are significant correlations for pretty much everything except for the neurofilament light chain, immediate recall and delayed recall. These are the only ones that don't have really statistical significant correlation, but total tau is quite interesting. It even has a positive correlation backwards digit span. How so? Actually, very interesting. This is expected. These are expected, but this is interesting. And also, every correlation, always correlation does not mean causation. We have to think with that in mind. All right. So, where does this one work? and they say that after all, the KIDE score is consistently linked to all three cognitive function measures, and notably this key genetic risk factor that they found in older populations, the E4 level of ApoE, was not associated with what they call neurotracker digit span, suggesting that the effects of it do not become apparent until middle to older age. And there were significant associations, as we saw, with ATN biomarkers, and as said, total tau was associated with lower immediate word recall, but not backward digit span scores. And although the higher levels of neurofilament light chain showed a trend toward lower backward digit span scores, the association was not statistically significant, as we observed. And a limited number of immune markers were significantly associated with cognitive function at ages 24 to 34 years, but some of these associations appear to become more robust in the following decade of life. So on the fifth wave of the study. These are the beta estimates for essentially all of the interleukin factors, all of the immune biomarkers. Again, this paper is open access, so you can just search up the name and you can find it. Yes, it is. I remember it is open access, yeah. Okay. This is repeats essentially what we said, what I said, and they said. They repeat themselves a lot in this article for some reason. I don't know why. Maybe I just haven't read enough papers. Maybe that is what the standard is these days. I don't know, honestly. But I've noticed that they repeat things over and over and over again, sometimes 3 to 4 times. Okay, so these findings provide preliminary evidence that ATN biomarkers may influence episodic and working memory a decade before middle age. So, to their knowledge, these are the first findings to suggest that ATN biomarkers are associated with cognition in a relatively large sample of 34 to 44-year-olds. As we said, several immune markers were associated with cognitive measures at ages 24 to 34, with this interleukin 6 and the HCRP associated only with backwards digit span. These are some more bit details about, you can read them if you're interested. I think I also included them. Yes, I included them in the in my notes. And we approach the end of this article. So getting after going through the bread and butter, we now have arrived at the limitations section. So they claim that this study has several limitations, and the significant associations observed between CRP and interleukin 10 in wave 4 were not present in wave 5. And they acknowledge that this may be due to differences in sample size, where the larger sample in wave 4 allowed for the detection of statistically significant but smaller effect sizes, and in wave 5 the effect estimates were closer to the null. And as previously reported, interleukin 8 in particular showed lower agreement between blood spot and serum assays. And this discrepancy might explain why there was an association between interleukin 8 and cognition in wave 5 was observed, but not in wave 4, which used but spot essays. Well, there is a possibility of type 1 errors. Sorry, let me just. I keep getting confused with uh, type 1, type 1, type 2 errors, and so on. I keep getting confused. I'm sorry. When the null hypothesis found significant differences, or there was no difference. Okay. Mm. So they acknowledge that there is a possibility of type 1 errors due to testing multiple models, but they did not correct for multiple comparisons because each model was designed to address distinct research questions that were determined a priori. A priori means beforehand. Um, mm, this is what we said as well. Immune inflammatory biomarkers already beginning to show associations with cognition pre-midlife. And as a wrap up, their findings suggest that key risk factors for Alzheimer's disease in older age begin to shape cognitive function decades before clinically detectable cognitive impairment and Alzheimer's disease, underscoring the importance of early prevention across the life course. So that was our stream for today. I hope you enjoyed it and learned a little bit. If you watch this later on, and I'm hoping I get some suggestions so we can review some more interesting stuff next time. And see ya.